Continuing on here with HIV and AIDS. So of course, HIV stands for human immunodeficiency virus, and that's the virus that causes AIDS, which is the acquired immunodeficiency syndrome. The bad thing about HIV is that it selectively infects helper T cells. These are CD4 plus cells. And as we saw in our study of the immune system, these helper T cells play a critical role in binding to antigen-presenting cells and then activating other parts of the immune system. If you take these cells out because they're virus infected or don't work anymore, or you simply don't have enough of them anymore, then you have an immune system, but nobody is calling it out. It's like being attacked by an enemy and your army and the Pentagon, everybody is asleep and nobody is waking up. So you have the forces available to fight, but nobody is calling them out to fight. And that's what the problem is with HIV and AIDS. So here a little bit on the origin of AIDS. It started to be zoonotic. That means it crossed over from monkeys to the human population. And that supposedly happened around 1908. It spread throughout Africa as a result of urbanization and increased sexual promiscuity. There's not that much information about it out there. Every now and then they're taking somebody out of their grave and checking for potential HIV that was present already. But so far, the first known patient that died from HIV is some person in the Congo in 1959. And then in the Western world, supposedly the first known case, some Norwegian sailor who died in 1976. So let's take a look at the structure of HIV. It's a lentivirus and it's a retrovirus. Retrovirus means that we have RNA as the genetic material and we're using reverse transcriptase to go back to DNA once the virus has entered the helper T cells. So we have two identical strands of RNA and we need the enzyme reverse transcriptase to make DNA from the RNA. So therefore we're going reverse from RNA back to DNA. And we also have this GP120 glycoprotein that forms the spike protein. So here's what the virus looks like. So we have here, this here is the virus. You have the RNA core, and then a protein coat surrounding that, and then we have an envelope. And in the envelope, you have the spike proteins, these little things right here. There's one that's enlarged over here. And so these are these glycoprotein spikes, the GP120 protein, and then the anchoring protein is the GP41. That's a transmembrane glycoprotein, and this is the envelope portion of the virus. So an HIV, as we said, specifically infects CD4 plus T cells. Those are helper T cells. Here's how the virus gets inside of your CD4 cells. So it is initially spread by dendritic cells and carried to the lymphoid organs and contacts active T cells. The GP120 just spike protein combines with the CD4 plus receptor on those cells that have it. And then you have some co-receptors, and together then you gain the HIV, the virus gains entry into the helper T cells. And releases its genetic material. So the virus will fuse and then enter into the cell, releasing the genetic material into the cell. So here's what this looks like. So here is HIV and the GP120 and GP41, the anchoring protein, right here, the spike protein. It latches on to the CD4 receptor. So those cells that have a CD4 receptor, that's where this virus can latch onto, gain entry. So you have viral fusion right here, and then the virus will release its genetic material inside of the CD4 plus cell, gaining entry, and then the fate of that poor cell is not good. So once inside of the cell, then the viral RNA is reverse transcribed back into DNA using the enzyme reverse transcriptase. Therefore, we call this a retrovirus. Then the DNA is integrated into the host cell chromosome and chromosomal DNA or genomic DNA. In an active infection, new viruses will bud from the host cell In a latent infection, the DNA is hidden in the chromosome as a provirus, but it may become reactivated at any time. Some of these cells become memory T cells that serve as a reservoir for HIV. One problem, a big problem with HIV, is that it undergoes very rapid antigenic changes and has a high rate of mutation. And the reason for that is that this enzyme up here, reverse transcriptase, is not that good in being accurate. So reverse transcriptase, in other words, makes a lot of mistakes, and these mistakes translate into mutations, and mutations translate to change. And that will be these rapid antigenic changes. And the high rate of mutation is due to reverse transcriptase not being a high fidelity enzyme. Here's a figure that shows you latent versus active HIV infection in CD4 plus cells. Here first the latent infection. There you just have HIV gaining entry, and it's going through the process of reverse transcriptase, and the provirus, the DNA, is integrated into the host cell genome and it stays latent, so we don't make actively new virus particles. But at any time you can switch over to active infection. In this case, the viral DNA here will be transcribed and translated. So transcription leads to mRNA, and then we get translation to protein, and we make more viral particles, more viral RNA, more viral proteins, puts the coat together, and then these new viruses, they will be budding off of the CD4 plus cell, and here you have new viral particles that will go off and infect more helper T cells. In this diagram, you have latent and active HIV infection in a macrophage. Basically, it's the same thing. So here is a macrophage is taking in the HIV particle, and we have integration into the host cell genome after reverse transcription, of course, of the viral RNA back into DNA, but you don't make actively new viral particles. If you have an activated macrophage, then this DNA here will be transcribed and translated into new viral genetic material and code proteins and spike proteins and whatnot. So whatever the virus needs, all the viral particles, they will be assembled, and then they will be budding off of this macrophage in this case, and then here HIV being released and going on to infect more cells. There are two main subtypes of HIV, and that's HIV-1 and HIV-2. HIV-1 is 99% of the cases. So these HIV-1 is related to viruses that infect chimpanzees and gorillas. That's the vast majority of HIV infections. And then HIV-2, they're related to viruses that infect monkeys and sooty mangabees. They're not often encountered outside of Africa. This one is less pathogenic than HIV-1. Okay, now moving on to the stages of HIV infection. Basically 3 phases that you should know about. Phase 1 is asymptomatic, but you may have lymphadenopathy. That means swollen lymph nodes. That means something's going on with the immune system. The immune system is fighting something. But it also could be asymptomatic. Phase 2, you have a steady decline of CD4 plus helper T cells. And this phase can last anywhere from a few months to a few years. And nowadays, actually, people are staying in this phase 2 with medication, apparently indefinitely. So you may in this stage have increased infections or persistent infections. You may experience fever or oral leukoplakia, and so you have a steady decline of CD4 plus cells. Once the amount of CD4 plus cells drops below 200 cells per microliter of your blood, that's when you end up with full-blown AIDS, and you enter phase 3. And this stage, you're going to have all sorts of opportunistic infections, and your immune system is basically shut down. Here are the phases of HIV infection in a diagram. So here the initial infection, you have high viral load here basically, but your helper T cell count is at first it takes a drop and then it comes back up as the body fights the infection. But then as you are entering from phase 1 into phase 2, you're going to have a steady decline of your helper T cell population. You're supposed to have about 800 or so. So this is your T cell, helper T cell count, CD4 plus T cell count. And you're supposed to have somewhere around 800, 900 of those cells per microliter of your blood. And there you go on a steady decline. And once you hit that critical number, 200 right here. So once you are hitting this number, 200 helper T cells per microliter of your blood, this is when you're going to have full-blown AIDS and you're entering phase 3, where you have lots of opportunistic infections. You're going to have yeast infections in your lungs and everywhere else in your body. You're going to have Kaposi sarcoma and a bunch of other symptoms. And they add up to AIDS, which is the acquired immunodeficiency syndrome, which is a collection of symptoms. The initial response to an HIV infection is usually strong, and you have a pretty effective immune response. So the cytotoxic T lymphocytes, they suppress the viral numbers, but once the HIV establishes a pool of latently infected CD4 plus cells, it's basically impossible to clear the infection, and that adds to the challenges of making a vaccine. There's also the impact of age on survival with HIV infection. If you are older or very young, and your immune system for some reason is not that good, then of course you're more susceptible and you end up with faster progression and more severe symptoms. There is an exposed but not infected population. Those are people with the CCR5 mutation, and we don't have time for this. And then there are long-term survivors, especially right now. There are some effective medications available that seem to be preventing people from dying. They don't ever go into stage 3, and so they are able to somehow maintain a sufficient CD4 plus T cell population. There are some diseases that are commonly associated with AIDS. You may have some protozoa type of diseases that most likely people would be fighting it off if they had a functioning immune system. But if you're immunocompromised, then protozoa can bother you. Cytomegalovirus, herpes simplex viruses, varicella zoster viruses are all much more severe in immunocompromised people. Mycobacterium tuberculosis, it's also a big problem with people that have AIDS or HIV infection. Fungal infections that normally wouldn't bother you because you have a functioning immune system, they could all cause big problems, including Candida albicans, so not so good. And cancers, Kaposi sarcoma, those would be a common cancer found in HIV infected people. As far as diagnosing HIV infection is concerned, the method of choice is ELISA, immunosorbent, enzyme-linked immunosorbent assay right here. You have to just keep in mind that the ELISA detects HIV antibodies, and that can take up to 3 months to develop those antibodies. So you need to wait for seroconversion. That's the period of time between infection and the appearance of antibodies. Within that time window, you will have negative tests, even though you are HIV infected, but the ELISA will not pick up because will not be positive because the antibodies aren't there yet. You can also do Western blotting, and then you can do plasma viral load that is determined by PCR reactions or nucleic acid hybridization. Now moving on to HIV transmission. HIV survives about 6 hours outside of cells. It can survive more than a day and a half inside of cells, and the typical routes of transmission include intimate sexual contact, breast milk, transplacental infection, mother to baby, contaminated needles, organ transplants, blood transfusion, and then anal intercourse. That is the most dangerous form of sexual contact as far as HIV transmission is concerned. Now let's take a look at HIV worldwide. So 35 million infected people worldwide, 70% of them in sub-Saharan Africa. The incidence typically is about 1 million cases per year. Heterosexual transmission now accounts for 85% of cases. Women are 42% of cases, 1/3 of cases in Eastern Europe, and Central and Southeast Asia are from injected drugs. And so here is sort of the distribution of HIV infection, AIDS, and region in the world. You can still see the hotspots right here. So in general, just Africa, you see the darker green here. There's some other places in Asia, yes, some South America, but in general, much of the world has fairly low, less than 1% of the population, but it's a huge problem worldwide still.